Good morning, good afternoon, everybody. So we will start our webinar today. Thank you for everybody joining us for our webinar. So my name is Susie Xueqin Zheng. So I'm the Assistant Director of the Asia Forest Research Center at UBC. So I'm responsible to coordinate the biodiversity and ecosystem specialized committee from Asia Pacific Asia Pacific Young Scientists Association. So today's webinar is organized by this association. So first, let me shortly introduce AYSA. And it was launched in 2020, aimed to helping young scholars with their professional and personal development and enhance the regional forestry research collaborations. So it is an open platform focusing on the Asia Pacific region. So it includes four specialized committees: forest resources management, biodiversity and ecosystem, and biomaterials and bioenergy and wood science and engineering. So if you haven't done it, please visit our website and register as a member. So I will post this link in the chat box later. So before starting, I want to inform everybody that the webinar will be recorded and it will be available on our online video platform. So for better sound quality, I will keep your microphone muted during the whole presentation. So in the QA section, after the presentation, you can unmute yourself if you have any questions for our speakers, or you can also post your questions in the chat box if you prefer. So today our keynote speaker is Dr. Huarong Zhang. He's a senior conservation geneticist at Kadoorie Farm and Botanic Garden in Hong Kong, SAR. He studied forestry for his bachelor from Beijing Forestry University and continue further his PhD in forest sciences, especially focused on conservation genetics from Northern Arizona University in the US. So after working shortly in the US, he joined the Flora Conservation Department at Kadoorie Farm and Botanic Garden in Hong Kong. In short, it's a KFBG. So he managed the conservation genetics laboratory and strive to translate lab data into useful information for natural conservation. Through cooperation with local and international partners, he has been engaged in developing conservation projects targeting key threatened species using cutting-edge genetic techniques. He runs various projects looking at genetic diversity of native species, as well as DNA and trade data to tackle the illegal wildlife trade. So he also co-supervise PhD students and the lab students interns from university in Hong Kong. So I know Huarong, Dr. Zhang long time ago. We both from the Beijing Forestry University. So we were like during the university time, we were both like members in the same student association and have a lot of common friends. So we also were colleagues at KFBG before. So today I'm really very happy that I can invite him to give the talk today. So please, Huarong, now it's your time now. Okay, I will stop sharing. Please go ahead, Huarong.
So
yes, we can see, yeah, we can see your PowerPoint now.
Okay. Sorry for the delay. So my name is Huarong. I am a senior conservation geneticist at Kadoorie Farm and Botanic Garden. And today I'm going to give a sharing talk tackling illegal and unsustainable wildlife trade with DNA forensics. So wildlife trade is a multi-billion-dollar business, and every year hundreds and thousands of plant and animal species are traded for food, for medicine, for furniture, for pets. So this is a major threat to biodiversity conservation, but also cause legal, social, and many other problems like human disease, COVID-19. It affects everyone. So it remains a huge challenge to tackle this. So it also requires a lot of efforts from government authorities, NGOs, and universities to work together. So today, I'm going to share our DNA forensic approach trying to tackle this issue. So my talk will have mainly three parts. In the first part, I'll use pangolin seizure as a case study to share how we study the illegal trade. And for the second part, so I'll share our market surveys. So using medicinal orchids as a case study to show how we study the market patterns. And for the third part, so I will share our shark fin monitoring project. So this is a collaboration project with Florida International University and Bloom Hong Kong. So why DNA forensics? When we survey the market, we found the market is quite complicated because we have seen plant roots, tree barks, tree leaves, animal horns, animal skins, animal parts, and even powders. So how do we know which species are made for these products? It's very difficult to identify. And also the wildlife trade is complicated. So we have seen one passenger carry pangolin scales, fly from Nigeria to South Africa, and then to Kenya, and then to Hong Kong. So basically, there are several different transits for this trade, and it's difficult to tell where are the true origins. The trade is also dynamic because it can shift species and also trade routes from time to time. So here is a general workflow for DNA forensics. So first, we have to take DNA samples very carefully. Usually we have to clean the surface of the samples to avoid contamination. And then we extract DNA and amplify them. So in many cases, those DNA samples are degraded. So we have to develop our own protocol to extract for extraction and amplification. Once we got DNA data, so we will compare the data with reference database such as GenBank, BOLD, and sometimes reference database is established by the scientists. And then we compare the data with reference data using statistical analysis to make identification for species and origins. So many species are threatened by the illegal wildlife trade, including some iconic species like elephants, pangolins, rhinos, bears, tigers. So we have special concern with pangolins because the pangolin seizures is actually increased a lot recently. So the seizures are found mostly in Asia and Africa, but also in Europe and other regions. So what are pangolins? Pangolins are mammals with a body covered with scales. So in this photo of pangolin, it's a injured pangolin, a Chinese pangolin found in Hong Kong, and it's in our rescue center receiving medical check-in. So there are 8 pangolin species, 4 in Asia and 4 in Africa. In Asia, we have Chinese pangolin, Indian pangolin, Sonda pangolin, and Philippine pangolin. Why pangolins are traded? So they are traded for meat because their meat are considered a tonic food in Asia and Africa, and also for scales. The scales are used as traditional Chinese medicine, but also used as traditional medicine in Africa. So Hong Kong is one of the main transit hub for the pangolin trade. So every year, tons of pangolin scales and also meat are confiscated in Hong Kong. So this confiscation represents great efforts from the authorities, but there are still many unknowns about these confiscations. For example, which species are behind these seizures, and where are these pangolins poached? So to answer these questions, we started to request samples from Hong Kong authorities and to study these seizures. So in 2013, we studied 2 large pangolin seizures from Southeast Asia. So at that time, the reference data for pangolins are still limited. So we collected DNA samples from Chinese pangolin in Hong Kong, and also we downloaded reference samples for the Sonda pangolin and the Indian pangolin. But the Philippine pangolin data is not available at that time. So the green color and the red color are the DNA we found from the seed samples from those 2 large confiscations. Most of the samples belong to the Sonda pangolin, but there are some samples we call scale H04 and scale H08. So these 2 haplotypes, they don't match with any of the reference data of Asian pangolins. So now we have DNA data for all of the 4 Asian pangolins. Still, these samples, they don't match any of the reference data. So it suggests these samples may belong to a new Asian pangolin species or a distinct genetic lineage. So this is worrying because some of these pangolins haven't been, maybe new species haven't been described in science, but already traded to near extinction. So from these seed scales, we saw a lot of genetic variation. So at least for 3 genetic groups. So basically, this result suggests these pangolins are collected from a large area. And these 2 seizures, so the white and red color, belong to the 2 different seizures. So these 2 seizures have very similar composition of this genetic diversity. So suggesting, so probably there's a criminal network, similar criminal network is organizing this illegal wildlife trade. So there are two main trade routes for the Asian pangolins. One is a land route from Indonesia to Malaysia to Indo-Burma and then to China and Vietnam. And another one is a sea route. So Hong Kong is on the sea route. The land route is more complicated, but not much study have been done on this route. So it may involve more pangolin species because in Hong Kong Seychelles, we mostly is from the Sunda pangolin, but from a land route, so there are probably more Asian pangolin species involved in the illegal trade. So from 2010, there are more and more seizures coming from Africa in Hong Kong Seychelles. So even though Africa is much further away, still the pangolins are still sourced from that region. So it suggests the Asian pangolins are probably more difficult to find in the wild, and now the trade is shifting to Africa. So we studied 30 pangolin seizures from Africa. These pangolins came in Hong Kong either by air or by sea. So by air could be El Pasos. So the El Pasos sent directly from Africa to Hong Kong, or sometimes via Europe to Hong Kong. And also they are passengers, so they carry pangolin scales in flights. By sea are those sea shipment in cargoes. So there could be tons of pangolin scales hidden in the cargo and shipped by sea to Hong Kong. So in these 30 seizures, we include both large pangolin scale seizures by sea from shipment and also small El Pasos. So we want to study these seizures to understand this trade. This graph shows the DNA result. We found all 4 African pangolin species in Hong Kong Seychelles, but mainly, so the number 1 trait species is the trait pangolin, or called white-bellied pangolin. So this species accounted for over 80% of the trade. And this species is probably more abundant than the other 3 species in Africa, and it is commonly found in the bushmeat trade and also medicine market in Africa. It's interesting because for the ground pangolin, we only found less than 10 samples in these seizures. So ground pangolin has different distribution than the other 3 pangolin species. It's mostly found in East and South Africa, but the other 3 species are mostly found in West and West Central Africa. So we don't know the reason why the ground pangolin is less commonly traded than the other 3 species. So this photo shows the details of the seizures. Although the tree pangolin is the most traded species, but you can see most of the seizures, there are more than 1 species, so not just the tree pangolin. So it's a mixture of different pangolin species. So this suggests the traders are actually mixing different pangolins together for the trade. So no matter it's tree pangolin or ground pangolin, so they probably collect whatever they can find in the wild. So origin identification is only possible for the tree pangolin because there are genetic studies to cover, to collect DNA data for this species all along its range. So this is a good opportunity for us to identify the geographic origin of the seizures, because we can compare the seized samples with the reference DNA samples collected before. So there have been 6 phylogeographic lineages described for this species. And when we compare the seized samples of the tree pangolin with the reference samples, so actually it also matched with the 6 phylogeographic lineages. So this result suggests actually the seizures are the pangolins are probably poached from all the range of these species. So, but mainly from Western Central Africa, and then followed by Dahomey Gap and Western Africa. So the poaching was widespread, but it concentrated in the Western Central Africa, which include Cameroon, DG, and also part of Gabon. So for this map, it clearly shows where are the reference samples was collected and where are the main poaching hotspot for this species. So you can see Western Central Africa is the main source region for this illegal trade, and we found a lot of genetic diversity in these samples. And if we look into the seizures further for the source regions, we can see actually most of the seizures have multiple source regions. Again, so this suggests the traders are putting the pangolins collected from different areas together, gathered in a central point for shipping. And also, the major export countries for this illegal trade is Nigeria and Cameroon. But from the source regions, we know actually these seizures are a collection of pangolins from different areas. So Nigeria is probably just a transit, not the source country. So we have shown the pangolin seizures by DNA forensic studies. So what about the legal market? So when we survey the market in Hong Kong for the dried seafood and medicine, we have seen a lot of plant and animal products. So we're quite concerned because we found a lot of endangered species in the traded products. For example, the medicinal orchids, Dendrobium crystollinum, and also the sea coconut from Africa for soup. It's also listed in CITES. And the turtle shells, emperor lute, shark fins, and many others like seahorses. So I'll give a case study of medicinal orchid we found in market. So Shihu is a traditional Chinese medicine. It has a long history in China, probably over 2,000 years. It's commonly found in Hong Kong market. So when we check the CITES trade data, there's not much species information available, but most of these dried stems are imported from Thailand. And when we check the customs data, so there's no trade code for this product. So basically, we don't know the species and origin, because when we survey the market, so the traders always say their products are coming from mainland China. So this is different from the trade records. We decided to use DNA to study this trade. So in the market, we have seen different forms of suhu for sale. So some are long stems, some are those coiled balls with different sizes, and some are large shoulder bulb, and some are small shoulder bulb. So this suggests there are different species or species groups used for this trade. And using DNA identification, we found over 20 species from market samples. And this photo shows the main traded species. So the number one is Danjobium divorialum, and then followed by Danjobium ophyllum. So these two species account for over 50% of the trade. The third one, Danjobium officinale, is a famous species, and it is widely planted in China. However, it's less commonly found in the market than Danjobium divorialum and Danjobium ophyllum. So about half of these species are actually not found in plantation, because when we check the plantation records in China, so mainly are these 8 Danjobium species, and number one again is the Danjobium officinale, and the second is Danjobium low-bellied. It's actually not found in the market samples, so this is interesting. Again, so the market samples identification shows different patterns with the plantation and suggests it's probably a mixture of planted orchids and wild collected orchids. So we mapped the traditionally used medicinal orchids and also the market samples in a phylogeny tree for the genus Danjobium in a published phylogeny tree. So because Danjobium has over 1,000 species, so this group is actually very difficult to work with. Most of the traditionally used Danjobium species concentrate on the northern group of section Danjobium. So the blue color in the phylogeny tree on the right. And also when we map the market samples, so it's in the red color. So you can see, so this tree has expanded a lot from the blue color to red color. So now I'm going to share our shark fin project. So this is a collaboration project with Florida University, Damon Chapman's team, and also with Bloom Hong Kong, Stan Xie. So every year, over 50 million sharks are killed for the fin trade. So, and Hong Kong is a major trade hub for this trade. Each year, Hong Kong imported shark fins from over 100 countries, but we don't know which species are traded and where are the major fishing regions. So when we survey the Hong Kong shark fin market, we found not only just those whole fins, but also the shark fin trimmings are also sold in the market. So shark fin trimmings are those trims between the shark fin and the meat. So this part is actually have different taste with the real fin. So usually it's sold at a low price. So for example, for one bag of shark fin trimmings, it's sold at about a few 100 Hong Kong dollars, but we can have over 100 samples just in one bag. So these treatments offer us an opportunity to study the market, because theoretically everything has treatments. So basically we can identify these fin treatments and find out the species traded in Hong Kong market. So how many species are sold in Hong Kong market for the shark fins? Using DNA, we find over 70 species. And the statistical modeling suggests we probably catch most of the species. And this graph shows the major traded species in Hong Kong market. So the number one is blue shark, accounting for over 30% of the trade. And the number two is silk shark. So this is the CITES listed species in 2017. And also the third one, scalloped hammerhead, is also CITES species. So we found a lot of endangered species in Hong Kong market. So this graph ABCD shows the families for these shark species, the habitats, the size, and also the conservation status of the species we found in the Hong Kong market. So about 1/3 of the species we found in Hong Kong market are actually endangered species. So this is quite worrying, because that means this trade really need to be checked to protect those endangered species. Because we sample this market every month since 2014. So this is a long-term trade monitoring project. And we want to know whether species composition is consistent across time to time. So we compared 3 years of species data. We found, again, those endangered species are actually quite common. So for example, silk shark, it's quite consistent in the 3-year data. It's ranked as number 2 in the market. And also scalloped hammerhead, smooth hammerhead are also commonly found in market. So when Stan surveyed the market, so he noticed that as a special trade for small fins. In the left photo, you can see the small fin is only about 1/5 of the regular fin. So they are whole fin, but they are so very small. So there are two possibilities. These small fins are from small-sized sharks. And the second, they are probably from juveniles of large sharks. So using DNA, we are trying to identify which species are involved in this special trade. And we found the most traded sharks is milk shark. So it is a small-sized shark. But the second one, scalloped hammerhead, is a large-sized shark. So from this species data, we found the small fin trade is actually a mixture of small sharks and juveniles of large sharks. So this is quite worrying. So because even baby sharks of those, including some endangered species, are traded for this fin trade. We also want to know where are the major overfishing hotspots for these traded shark fins. So I'll use scalloped hammerhead as an example to show how we identify the source regions. So scalloped hammerhead is commonly found in Hong Kong market. It has a wide distribution across global ocean waters, but it is a critically endangered species because its population has over 80% decline in the past 20 years. So the reference DNA data has been collected for this species along its range, and as a genetic signal found between different populations. So this map shows the main 9 populations for this species. So you can see from Central Pacific to East Pacific to Northwest Atlantic, East Atlantic to Indian Oceans. So this species has a wide distribution, but we can identify these 9 populations using DNA. And Andrew used genetic stock identification analysis to find out the source populations of this species sold in Hong Kong. So actually, 6 out of 9 populations can be found in Hong Kong market, but East Pacific is a major source population for this species for the traded shark fins found in Hong Kong market, and then followed by West Pacific and the Indian Oceans. So to summarize, we found complicated trade networks, both in illegal trade, but also in the legal market. And the species sourcing have been expanded. So like in the case of medicinal orchids and also shark fins. So they included a lot of species in the trade. And the trade routes and the collection networks suggest they are probably organized criminal networks behind this trade. But using DNA forensics, we can identify the major traded species and also the poaching hotspots to support law enforcement and also conservation of the endangered species. And finally, I think public education is a key to help reduce the demand and tackle the illegal and sustainable wildlife trade. I would like to thank Hong Kong customers and AFCD to donate pangolin samples to us for study. And I would like to thank our lab staff and the KFBG colleagues and also our collaborators to help with these studies. And with that, I would like to say thank you, and I'm happy to take questions.
Okay, thank you, Huarong, to offer us a great talk. So a lot of information. I think for me, it's really very impressive. So now we will open the question section, and so I will take the advantage as a host. So I will first ask 2 questions. The first one, so maybe we can switch to a more easier like topics. And so for it's not a question from me because I have worked in KFBG before. So, but for most people, I think people really probably have some doubts that why KFBG is a botanic garden, why they have conduct so many wildlife studies. So yeah, could you offer us some background, like history information about this?
Oh, sure. So the full name of KFBG is Kadoorie Farm and Botanic Garden. So it started from a farm in 1950s, and then right now it's more biodiversity conservation focused. So we have flora conservation for plants and also fauna conservation for animals. So we have an animal rescue center in Flora. And also we have Kadoorie conservation in China. So mostly focused on China conservation programs and also education program for public education. So those are the main conservation related programs in KFBG.
Okay, thank you for the information. Yeah, so it's yeah, as I also like working candle farm and botanic garden before, so it's like a really quite diverse like a institute or research institute, also like education organization. So they really put a lot of effort like on the conservation part, like for the wild plants and wildlife, yeah, everything. So I have another question. Like you introduced like a lot of DNA like data for the for trade the like illegal wildlife or wild plants trees, and I'm wondering, like, is there like any rapid like DNA tools which can really use for in for the customs, like Hong Kong customs, to really immediately like break or identify the illegal like wildlife or wild plants trade?
Yeah, that's a good question. So there are DNA techniques available now to check identification in a few hours, like real-time PCR. So basically, for those endangered species, you have already developed protocols, and then with real-time PCR, you can find out the identification results just probably in 1 or 2 hours, so you can get the result. So one of our collaborators, Diego, already tried real-time PCR in Hong Kong customers to check those imported shark fins. So it's very helpful. So basically, you can get a result just in a few hours. And it's also very powerful to check those protected species.
Okay, great. Okay, so like yeah, for everyone, if you have any question, please like unmute yourself and yeah, just ask our case speaker directly. Professor Wang, do you have any question?
Yeah, thank you so much, Dr. Zhang. I have two questions. Thank you for your excellent presentation. And the first question is about like technology for DNA like monitor checking. So is it the mature technology? Also, do you have enough database to check all the import like wildlife species in Hong Kong, and if yes, what kind of cost associated with that kind of checking? The second thing, I was just wondering why Hong Kong always like a center for the illegal trading. Why is I don't know what the legal or policy issue or political issue or law issue was always is all not only for wildlife, for wood, for all those kind of things. Hong Kong always the illegal like center, trading center. Do you know? This is my question.
Right. Okay. So for your first question, DNA techniques have been developed to check like plant and to identify plant and animal species for decades. So initially there was just probably in a database, not many reference data available. But right now, with initiative to barcode the living organisms on the Earth. So there are more and more reference data available at a species level. But for provenance identification, it's more difficult. For example, for pangolins, we still cannot identify most of the traded pangolin species. Right now, it's only possible to roughly identify the source regions of the trade pangolin. So as I have showed in my presentation, but for the other pangolin species, it is still not possible because it requires reference DNA data along the main range of the species. So it's quite challenging. But I think more efforts is going on right now to establish the database. So in the future, I hope we can use the DNA data to find geographic origin for more traded species. So for your second question, so Hong Kong is actually not just the trade hub for these endangered species. Actually, there's a lot of shipment trade going on. So a lot of goods coming through Hong Kong because Hong Kong is a main transit for these goods. It's kind of like a global transit for all the goods, not just for wildlife. So, but in this trade, so wildlife trade is quite significant. So when we check the trade data, especially for like pangolins and sharkfins, like for example, the sharkfins, Hong Kong does have a big market, but also part of the trade goes to other places. So basically, it's both a transit and also a destination market. So that's why we use shark fin study in Hong Kong market as a case to actually study the global fin market. So because historically, Hong Kong market is an important market, but also it's also a transit for this trade. So for pangolin scales, so Hong Kong is mostly a transit because we cannot find pangolins sold in Hong Kong market.
Okay. Okay, thank you so much.
Okay, so is there any more questions? Okay, so like there's one question from the chat box. So Liu Jie want to ask, which DNA marker do you use? I think probably different, also different species, you use different marker.
That's right. Yeah. For example, for the pangolins, we have used the CO1 and CYTB and also D-loop. For the sharkfins, for species identification, we used CO1. So it's a short fragment developed by Damon's team in CO1. But for origin identification of the hammerhead, Andrew used the D-loop region. But for plants, so for medicinal orchids, we use both ITS and PSBA. So actually, yeah, for different species groups, we use different markers. Especially, we also it's the purpose is for species identification or for origin identification. So we also use different markers.
Okay, that's good. So is there more questions for Dr. Zhang? Okay, yeah. If not, maybe I will just give like a short summary of today's webinar. So it's a really great talk. So including a lot of information, also very impressive. So about the like DNA technology in wildlife and wild plants conservation. So except this scientific knowledge, it also reminds us to need to pay more attention in our daily life to protect our precious species. So biodiversity conservation is not only a scientific topic, moreover, it should become a like code of our behavior. So no trading, no killing. That's really very serious. I thank you, Professor Dr. Zhang, for your talk. Thank you everybody for joining us today. Yeah, so sorry for the technology problems. Like, yeah, we had some breaks in the beginning. And Professor Wang, you want to say something?
No, thank you. Just want to say thank you. Thank you so much for your excellent talk. And we definitely learned a lot. Thank you so much.
Yeah, thanks for the invitation. And I apologize for the delay. Sorry about that.
It's happened all the time. Yeah, thank you so much. Yeah.
Thank you.
Okay, yeah, thank you. So in the end, I would like to remind everybody. So AYSA, we will host our webinar regularly. So the details will be announced on our website and sent by emails to our members. It is open to public, so you're always welcome to join our webinar. Okay, thank you. Thank you, everybody. See you next time.